So, what is the causal pathogen of Fusarium wilt? Fusarium wilt of banana or Panama disease is one of the devastating soil-borne fungal pathogens that caused by Fusarium oxysporum fsp cubans (FOC). The fungal produces three types of spores, which is microconidia, macroconidia, and chlamydospore. On PDA media, the colony appears as pink cottony. The microconidia has 1 to 2 kidney-shaped structures, while the macroconidia has 4 to 8 cells enclosed in sac-like structure, which is sickle-shaped. Now, we will look on the disease cycle of FOC. Firstly, the FOC spores present in the soil adhere to the secondary or tertiary feeder root caps 1 to 2 days post-infection. Then the spores germinate upon stimulation of root exudate, followed by the root invasion through hyphae penetration into the cell wall or wound. As FOC invades the vascular tissues, a large amount of FOC spores are constantly produced and rapidly distributed throughout the whole plant via the host transpiration system. Then, the hyphae clog the xylem vessel. As the water uptake is restricted, the progressive wilting of all the leaf and the collapse of that leaf form a skirt around the split pseudostem. The FOC feeds saprophytically on the dead tissues and produce chlamydospores that remain viable for more than 30 years and will result in another wilt outbreak upon direct contact with susceptible banana's root. Now, we will look on several management strategies to control the disease. The first management is through mutation breeding and transgenic approach. Malaysia has successfully produced a FOC charpor-resistant mutant from Rastali, also known as Rastali mutara, derived from a rigorous selection of susceptible Rastali in infested fields. Meanwhile, the transgenic Rastali enanca genotype have enhanced disease tolerance under glasshouse conditions, but no first trial validated the consistency of this promising line. Second is intercropping. Intercropping with Allium tuberosum can reduce disease incidence in certain types of banana plants. The plants from Allium family were largely utilized in intercropping due to their production of antifungal volatile compounds.